Good morning, everyone, and welcome to today's webinar sponsored by the HIBAR Research Alliance, also known as the HRA. I'm Scott Slovic from the University of Idaho, and I'll be the moderator of today's one-hour event. This is the third in our series of webinars for researchers, research administrators, and others who are interested in what we call highly integrative, basic, and responsive research. The HIBAR Research Alliance provides a website that describes the activities of the organization's 4 collaborative action groups, or CAGs. CAG number 2 is working to encourage the creation of new HIBAR research projects. And it's with this goal in mind that we've launched this webinar series comprised of presentations by experienced HIBAR researchers and administrators. Our first 2 webinars are now available for viewing on the HRA website, which can be found at hibar-research.org. Before we start with today's featured presentation by Dr. Ron Hardy, I wanted to explain the structure of today's meeting. We'll begin with a brief introduction to HIBAR. Next, our featured presenter, Dr. Hardy, will give us an overview of the HIBAR aspects of his research for approximately 20 minutes. We'll then have 15 or 20 minutes for Q&A, followed by a brief opportunity for Dr. Hardy to offer any final takeaways. And now I believe we're ready to get started. So I'd like to begin by introducing Dr. Lauren Whitehead, Professor of Physics and Special Advisor on Innovation, Entrepreneurship, and Research at the University of British Columbia, to offer an introduction to the concept of HIBAR and to the HIBAR Research Alliance. Lauren?
Thank you very much, Scott. We've got just a few slides to show, and I'll be relatively brief with them. Well, good morning, everyone. I'd like to just very briefly speak about the HIBAR Research Alliance and HIBAR research. The reason, of course, is that we're about to hear a very interesting presentation from Dr. Hardy on his research, which is a beautiful example of this idea. So the topic here is research types, and it's a bit philosophical, but if you think of research, it ranges from work that can be very highly theoretical to work that is extremely practical. And there's, of course, a spectrum in between. So we have various labels for research, and a very common label, for example, is basic research. And if you think of what basic research feels like, it tends to be work that is more toward the theoretical end of that spectrum. But of course, it has various flavors as well. And then similarly, applied research projects have a different distribution, where most of the work, most of the time, is much toward things, more oriented toward things that happen more quickly and are practical. So these are obviously very well-known terms. And the term HIBAR research applies to kind of that middle ground sketched in this diagram, where the idea is that it's middle ground from a time perspective, but also in many other ways. And as you'll see in various information that our group is providing, this kind of research, while it's common, is not as common as many think it should be, because it tends to be highly well-oriented toward creating interesting breakthroughs. So the mission of the HIBAR Research Alliance, a group of 11 universities, as Scott mentioned, is to achieve, within 10 years, a system-wide 4-fold increase in the fraction of research projects that are HIBAR. And this is throughout the entire university system, not just within the 11 member universities. And we'd like that to translate into what we think today is about 1 project in 20 having this HIBAR character, which you'll hear about today, to about 1 in 5. So we think it's possible and desirable for that to become more common, while also strengthening all other important forms of research. So that's what the Alliance is about. And in today's talk from Dr. Hardy, I guess I would suggest that you might be on the lookout for the 4 characteristics, or we call them sometimes the 4 dualities, that make up HIBAR research or identify HIBAR research projects. And I'll just describe those very quickly with regard to Dr. Hardy's project, and then hand over back to Scott. The first duality is one of motivation. So in the project you'll hear about today, there's very basic work on fish genetics, but it's also oriented toward a critical world problem, food security. So the motivations of solving problems and deep understanding simultaneously is one of those characteristics of HIBAR. Second one is dual methods. And what we mean by that is a combination of typical high-level academic approach to understanding, combined with creative approaches to solving problems, which is often not part of the standard academic approach. Also related to that is the idea of involving people from outside the academic system. And so academic researchers really working in deep integration with experts in outside organizations is typical. And that certainly has been the case with Dr. Hardy's work. And then lastly is kind of, I guess you could call it an intermediate time frame, where the work itself is deep and is often associated with long-term knowledge objectives. And yet at the same time, there's a sense of urgency, a sense of actually having the results applied to the real world. And I think you'll agree with me that Dr. Hardy's story is a beautiful example of that. So Scott, on that note, I'd like to hand back to you.
Thanks very much, Lauren. And now I'm pleased to introduce Dr. Ron Hardy from the University of Idaho. Dr. Hardy is a university distinguished professor in the Department of Animal and Veterinary Science, where he's an expert on animal nutrition, specifically fish nutrition. He was a professor in the School of Fisheries at the University of Washington and a research scientist at the Northwest Fisheries Science Center in Seattle before he moved to Idaho in 1996 to serve as the director of the Aquaculture Research Institute. Today, he will tell us about his longtime work in the field of fish nutrition and breeding, conducted with colleagues from throughout the world and from US government agencies and industries, leading to fundamental discoveries about fish digestion, gene expression, and microbial communities in selected trout, while also responding to the global demand for food security. So please welcome Ron Hardy.
Okay, well, thank you very much, Scott. I'm going to share my screen now. Thank you very much for the invitation to share our story here at the University of Idaho. And as you were told already, I'm going to talk to you about HIBAR research in aquaculture, which spans the spectrum from very applied research to basic research. Just before I begin, I will say that aquaculture per se is really a form of food production. It's not necessarily an academic discipline, but it's a discipline in which various other scientific disciplines contribute to its advancement, such as fish biochemical nutrition, genetics, fish disease and immunology, and others. With that, I will go on and begin, I guess, with a brief description, just for those of you that are unfamiliar with aquaculture. It's defined as a farming of aquatic organisms, and these include fish, both freshwater and marine, shellfish, crustaceans, crabs, and shrimp. In many parts of the world, other animals, turtles, frogs, sea cucumbers, and all there are over 300 species of animals, plants, and shellfish that comprise aquaculture worldwide. And at the moment, it is the fastest growing livestock animal production sector on Earth. In the little diagram below, you can see the black line depicting capture fisheries, wild fish capture, and you can see that somewhere in the late 1980s and early 1990s, our yield from capture fisheries pretty much leveled out. We're fully exploiting the fisheries research from the sea. But over the same period, you can see in the red bars the growth of aquaculture, which has made up the difference between supply and demand to supply seafood for basically the consumers of the world. Aquaculture, we think of it as a new enterprise, but it's not. It's been around for at least 4,000 or 5,000 years, documented in Rome or in Italy by the Etruscans, the Chinese, and others. The oldest record was a record of a man, and shown in the right, his image, named Fan Li, who wrote a carp farming manual. It was recorded in the 5th century BC. His system of farming was based on fertilizing ponds to produce natural food to grow the fish. Production was pretty low, about 200 to 300 kilograms per acre, but it was a very effective system used mainly in Southeast Asia, but elsewhere. And crazy as it sounds, for the next 2,500 years, fish farming basically followed this system. Fertilized ponds, rely on natural food production, and use that to produce fish. Beginning though about 90 years ago, aquaculture shifted and began its new phase, which involved supplemental feeding of fish. And that is kind of characterized by a shift from traditional aquaculture, which I just mentioned, to industrial aquaculture. It's shifted from basically family-run farms, small village farms, to more industrial farming characterized by monoculture of species as opposed to polyculture. and much greater inputs in terms of energy, feed, and other factors. It now supplies, aquaculture does, about 50% of all the fish that are consumed in the world. But looking ahead, population growth, increasing incomes in developing countries will continue to increase demand for fish. And as I mentioned, captive fisheries are fully exploited, so there's no really increase that we can foresee in capture fisheries. So fisher demand, as I mentioned earlier, can only be met by increasing aquaculture production. So our challenge is, how do we do this in a sustainable fashion? We need to work ahead to develop sustainable feeds. Worldwide, freshwater resources are fully utilized and spoken for, so there's very little room to increase freshwater production of aquaculture other than by intensification. So what we're seeing is a move to RAS systems, which I'll define in a minute, but are land-based recirculating aquaculture systems that use water over and over again. And then the other challenge that we face is environmental impacts, reducing some of the waste product release from farms and some other factors. HIBAR research has enabled this aquaculture growth when you think about it. 4 factors that have really stimulated aquaculture growth include development of vaccines against diseases, advances in understanding of fishery production and larval fish rearing, development of new technologies, but most importantly, from my perspective, being a fish nutritionist, is development of effective feeds that supply all essential nutrients fish need for growth and health. And that really began to have an impact somewhere in the mid-70s. As you can see by my little arrow there, following the development of scientifically formulated feeds, aquaculture grew substantially, primarily in Asia, but also substantially in other parts of the world. This was enabled by a main breakthrough in the 1950s that led to new ways to identify nutritional requirements of fish. Prior to that period, fish feeds were made by formulating them empirically, basically trying ingredients, taking them out, putting new ones in, and measuring the production of fish. But with the identification of the nutrient requirements of fish, for the first time, feeds could be formulated to be nutritionally complete. What this has done is enabled intensification of production, going from 200 to 300 kilograms per hectare in the past to over 1,200 and even more in some cases, kilograms per hectare. This has been depicted by many people, including The Economist, as the blue revolution in food production, to contrast it from the green revolution in crop production. The outcome of this has been to increase food security for over 3 billion people, and aquaculture becoming a major international business. For the most part, up until recently, and in the last 10 or 15 years, production feeds used fish meal as their primary protein source, which fish meal, as you may know, is just dried whole fish powder made from species that people don't consume directly. 5 to 6 million metric tons are produced annually, but by 2007, the increased demand for fish meal and fish feeds exceeded the global supply, making it necessary for aquaculture feed manufacturers to turn to alternative proteins from grains and oilseeds. And the challenge for us as fish nutritionists is that most of the fish that we highly prize and are high value fish are carnivores. How can we turn these fish into vegetarians? It turned out replacing fish meal was much more challenging than we imagined. Fish require about 40 essential nutrients in their diets, similar to land animals and poultry. And the nutritional content of plant proteins is very different from that of fish meal. For example, plants don't have bones. So the content of essential minerals in plant proteins is much different. Fish meal contains long-chain, highly unsaturated, polyunsaturated fatty acids, which are essential nutrients for fish. Plant oils do not. And most importantly, the amino acid profiles of plant proteins differ from those of fish meal. So what we found when we start substituting plant proteins for fish meal was growth slowed. We had to feed more feed per kilogram weight gain, and this was related to increased protein turnover. We also saw some nutritional deficiencies in production fish that we hadn't seen before, including lens cataracts, spinal and jaw deformities, impaired retinal development. So fish couldn't see very well. And you can see some examples on the right of vertebral skeletal deformities, namely scoliosis and lordosis, and other pathologies, including intestinal enteritis, inflammation of the intestine. So the conventional applied research approach, not HIBAR particularly, is to basically replace fish meal and land animal proteins in the diets with plant proteins, which we know this changes the amino acid profile of the feeds. So the solution, following the leads of livestock and poultry nutritionists, was simply to add supplemental amino acids to meat the dietary requirements as we knew them. And the outcome of that was improved fish performance, but not equivalent to fish meal diets. And basically, the more we added, the more we replaced fish meal, the more we had an impact on growth. Looking at this from a larger perspective, we have been following for a very long time something called the law of the minimum, also known as Liebig's barrel. Now, Liebig was a botanist in 19th century Germany who made the discovery that the growth of plants was controlled by the minimum level of an essential nutrient. So he created what we call Liebig's barrel to illustrate this graphically. And the height of the staves of the barrels illustrate the level of each individual nutrient. And so the lowest minimum level of an essential nutrient, depicted over there in the barrel under minimum, would be the limit that the plant could grow, even if the other nutrients were there in excess. And this led to the concept we use today in human nutrition, and that of a minimum dietary requirement, and what we use in animal nutrition as the first limiting amino acid, and meaning lysine. And this really has been how we've looked at things for over 100 years. So the law of the minimum would tell us that fish meal has an ideal balance of amino acids for fish, and plant proteins have an amino acid profile that's deficient. So, for example, corn or maize protein is low in lysine, soy low in methionine. So if we correct this by supplementing the feeds with crystalline amino acids, we should be good to go. And no one's really thought much about the levels of other amino acids that may or may not be present at required levels, or sometimes in excess. This approach has been based on, which we don't always really think about or understand, on two key assumptions. And that is, all we need to do is meet the minimum essential amino acid requirements of fish or animals, that digestible nutrient levels are interchangeable between ingredients. So nutrients are nutrients, is what people say. And if nutrients are digestible and the levels are correct, all should be well. And just to define digestibility as we define it in animal nutrition is the difference between ingested nutrient levels and the levels of nutrients that are excreted in feces. The assumption being that the difference is the amount or value or the percentage that is absorbed in the fish and utilized for growth. And looking at this initially, we measured digestibility of different plant proteins and different diets containing these plant proteins. And we find that digestibility of protein and amino acids is really pretty much the same. In fact, even sometimes quite a little bit higher in plant protein diets. So that doesn't really get us where we want to go, nor provide the insights to explain the differences in growth that we see. So our HIBAR approach that we've utilized here at the University of Idaho has been a university government partnership that I created 20 years ago through congressional appropriations with the U.S. Department of Agriculture, Agricultural Research Service. Our goal in this collaboration of the formal partnership was to initiate a program of selective breeding to develop a trout strain that would flourish when fed a plant protein diet. And so we've now been doing this for almost 20 years. We're in our 8th or 9th generation. After selection and very heavy selection, basically, to identify family lines that exhibit higher growth performance when they're fed plant protein diets. Our selected trout now grow twice as fast, and it doesn't matter whether they're fed fish meal or plant protein diets, but they grow twice as fast on plant protein diets compared to non-selected fish. And it dawned on us that this is highly relevant to HIBAR research, because the selective strain, in addition to being valuable to aquaculture, it's also an interesting or perfect model to allow us to explore physiological and genetic mechanisms that are responsible for the increased performance we see in these fish. So our approach has been to compare the model trout with non-selected fish, and initially looking at the question of why they can do well on plant protein diets has involved looking at temporal differences in amino acid uptake following a single meal, look at amino acid signaling to see what effect that has on tissue protein metabolism, measure the activities of amino acid transporters in the intestine that actually carry the amino acids into the tissues, and then also characterize the trout, the microbiome and the effects of trout strain and diet on that. So that's what I'll show you here briefly. So in terms of the physiological approach, looking at plasma amino acids, our approach was to force feed a normalized feed quantity to fish and measure plasma amino acids over a period of time in the portal vein and the caudal vein. So the values in the hepatic portal vein provide insight to us to show amino acid absorption in the intestine, whereas the caudal vein shows the availability or what the peripheral tissues actually see, but also gives us some insight into catabolism and protein turnover as well. We also, we fed this plant protein blend that we created with or without supplemental amino acids, plus we looked at the 5 common ingredients we use and to try to determine what are normal patterns and what are different patterns that we see when we feed the plant proteins. So we sampled blood from 2 places, the hepatic portal vein and the caudal vein after a single feeding. And to give you an example of, we have tons of data on this, but to give you an idea of what we see in the hepatic portal vein, looking at the selected strain shown in the gray line and the non-selected strain when amino acids are not fed, is we see in the selected strain a slight increase and a normal decrease as tissues, as the amino acids are absorbed. But interestingly enough, in the non-selected fish, we see 2 peaks. We see an initial peak after 6 hours of feeding and another one at 18. Looking at the difference between strains, we see a difference initially with the non-selected fish showing the increase I described a minute ago when we add amino acids, an increase at 12 hours. But interestingly enough, we see a much higher level at the early hours of absorption and then a gradual and what we would like to see, which we think is a normal pattern of consistent and gradual decrease in the hepatic portal vein, meaning that absorption of these amino acids in the selected fish is a nice, smooth pattern over time. So to summarize this, without amino acid supplementations, the difference between strains are minimal, even though we have 2 peaks in the non-selected. But when we add the amino acids that we commonly would supplement, we see significant differences. This gives us evidence of amino acid signaling in the selected strain influence digestion. And the later peak in the non-selected fish suggests elevated protein catabolism in the peripheral tissues. Looking at the caudal vein, and just to remind you, this is what the tissues see and what are used for actual protein synthesis. Comparing the 2 strains, when amino acids are supplemented to the diet, we see the same type of story with the selected strain, the blue line, showing a gradual decrease and a leveling off over the 24-hour period. Whereas in the non-selected strain, we do see a leveling off up until about 12 hours, but somewhere in the 18 to 24 hours, we see an elevation. This suggests to us tissue catabolism in peripheral tissues. So just to summarize the findings from this part of our HIBAR research, we find that supplementing amino acids to plant protein diets alters the uptake pattern of not just the ones that we supplement, but all of the essential amino acids. We see the selected trout phenotype exhibits different patterns. And the plasma tells us then that these plasma amino acid level measurements are a potential valuable tool to help us assess ingredients and to look at the effects of processing that are being employed to hopefully improve their nutritional value. Also, it allows us to evaluate formulated feeds and other supplementations. As I mentioned earlier, we've also looked at amino acid transporters in the intestine and at the microbiome to briefly describe our transporter work. We do see some differences in amino acid transporter levels. Here we're measuring gene expression that were affected by dietary treatment, particularly in PEP1, which transports small peptides and free amino acids. So we do see some differences there associated with strain and diet. In the microbiome analysis, we see the non-selected strain microbiome is altered significantly by feeding plant protein diets, and it tends to reflect the microbiological community that's present in the feed. And this is compared to a fish meal feed. But the crazy thing and the interesting thing we found was that the selected strain microbiome was not altered by feeding the plant protein feed. It really resembled the fish meal feed. And this is something we're exploring now in more detail. So the HIBAR approach has really proved valuable to us. It's altered some long-standing paradigms that we've accepted for 100 years, further exploration in animal and fish nutrition. And number one being nutrients are nutrients. As I mentioned earlier, nutrients assume the different sources are interchangeable, but we see that that's not the case. Digestibility predicts performance was another pretty much assumed reality. We see now that that's not the whole story because Digestion timing is crucial to synchronize amino acid levels for optimum protein synthesis in the muscle. We found evidence of amino acid signaling that affects cell metabolism and protein retention. And we also found that ingredients alter gut function directly and also indirectly by altering the microbial community, which has implications in terms of immunity. And also the microbes provide some essential nutrients to the fish directly themselves. So the impact of this really changes the picture as far as we are concerned. Supplementing amino acids to balance the diet doesn't necessarily correct the problems we see with low protein retention in plant-based diets, at least in non-selected strains. But our model fish, our selected strain has overcome these problems and grows very quickly. So the HIBAR approach shows that our selected fish strain is a unique model to explore novel aspects of amino acid metabolism, and that research will continue with our partners. I'm going to switch gears here very quickly and just give you an example of another one of our HIBAR research activities. This is being done with tribal collaboration with a consortium of 4 Upper Columbia River tribes that are interested in restoration of salmon populations in areas where they've been extirpated. And one of the considerations there is thermal tolerance of fish. We're focusing this work on red band trout, and these fish exist in the yellowish area in the map to the left, which you'll recognize as being inland. These fish exist in desert areas where water temperatures and upper temperatures in the summer get very high, and also in montane areas where the maximum temperatures are much more modulated, as illustrated in the little figure on the right. We measure thermal tolerance in these trout ecotypes by actually conducting EKGs to measure cardiac function. We attach electrodes to the fish, and you can see, electrode, you're probably familiar with what EKG results look like, but under 15 degrees, that's a normal EKG showing regular heartbeats. Each little peak is 1 heartbeat. As we increase the temperature to 20 degrees in the fish, the number of heartbeats increases. And finally, when we continue to increase the temperature, we reach a point where the fish go into arrhythmia, arrhythmic heartbeat. We define that as the critical upper thermal maximum. And looking at the figure in the lower left, what we found in our research is the fish from these different environments, these ecotypes, exhibit different CT max, critical thermal maximums, with the desert fish that are exposed to the higher temperatures being able to tolerate higher water temperatures before they reach CT max compared to the other 2 ecotypes. And just to show you what that means in terms of number of heartbeats per minute, in the figure on the lower right, the yellowish high line is the desert fish, and you can see that they continue to increase their heartbeat even up into the upper lethal range of temperatures, close to 30 degrees centigrade. Whereas the other ecotypes that have evolved, or at least inhabit cooler water, do not possess this capacity. Their heartbeats drop off. They reach their arrhythmic level somewhere in the 24 to 27 degree range. This is pretty interesting to us. So we're interested in looking at some of the, if we can identify single nucleotide polymorphisms that are associated with these differences in cardiac function, and showing you just an example of the kinds of the ways in which we portray this, showing the desert on the top, the in between cool montane fish and the cold montane fish. We're seeing some interesting peaks on chromosome number 6. We're now expanding that and seeing if we can identify what these genetic sequences are and what they code for. So that will give us some insight in some of the metabolic differences these fish may possess that enables them to perform as they do. So as far as the technical impacts of our HIBAR research, as I may have mentioned, global supplies of fresh water are fully exploited. We see going forward over the next 5 to 10 years a shift of fish production to land-based recirculating aquaculture systems. These systems are different from flow-through or ocean systems. So we see a need to develop strains of fish that can flourish under these RAS rearing conditions where water quality parameters such as temperature and dissolved oxygen, suspended solids will be different. And we hypothesize that the SNPs that we're seeing in our cardiac function studies are probably related not just to cardiac functions, but to overall respiration and oxygen transport, which will be critical characteristics of fish that we will be looking for to populate these systems. We also think these SNPs will be useful as markers for selective breeding of fish, not just the fish we work on, which is mainly trout, but other species of farm fish, of high value fish. As far as HIBAR societal impacts, identifying of mechanisms by which these fish adapt to environmental challenges will help us not only in aquaculture, but also in our ecological studies, particularly with the tribes, to identify strains of fish that would be appropriate for reintroduction into areas with specific thermal water temperature profiles. We're very interested also in the phenotypic plasticity of fish populations, how they're able to adapt with environmental conditions. And of course, I mentioned already the application to RAS production, land-based salmon and marine fish production. In addition to reducing pressure on water resources, there are other environmental concerns that are quite evident to those of us in the Pacific Northwest who are familiar with some of the controversies surrounding salmon farming. And this includes waste, uneaten food and fecal waste from pens, escaped fish, interactions with wild fishery stocks. Another societal issue is the conflicts that exist between recreational uses of coastal lands and those used to raise fish or food producing uses. But the other societal demand is, as we go forward, we need to ensure safe, healthful, and traceable fisheries products for consumers. We think land-based farming has the capacity to contribute greatly to this, to let consumers know where the fish come from, what it's been fed, the conditions under which it's been reared, and this will improve the perception of consumers anyway with regard to farm fish. So to wrap it up, our work has global impacts as we see them. Our work, HIBAR research, will contribute to the development of sustainable plant-based feeds, both for carnivorous, freshwater, and marine fish species, moving forward with fish strains that will increase productivity and overcome some of the problems we see right now with recirculating aquaculture land-based systems. And then at the bottom line, basically, our big goal as we think down the road is improved food security for future world population of 9 billion people. So thanks very much.
Thank you very much, Ron. That was such an informative and interesting and important presentation. Great. I see that we have 2 questions that have come in from attendees. So let me begin with those. We have approximately 20 minutes for questions and for any final takeaways you'd like to leave us with, Ron. So one of our attendees is asking that you mentioned you've been working on this project for more than 20 years. How do you maintain enthusiasm for everyone involved in the project for such a long period of time?
A lot of our studies, as you can imagine, are incremental. We didn't start out 20 years ago, for example, with our selective breeding saying, okay, here's an endpoint. We're going to be, as soon as we achieve improved strains of fish that can tolerate plant-based diets, we've met our objective. We continue to see other opportunities for selective breeding, looking at the effects of our selection program on fish health parameters, how they react to other functional feeds. Basically, we don't see these projects as having a discrete end because new problems emerge, new issues are brought up. And so we basically, as all researchers, create hypotheses, answer our hypotheses, and often end up with more questions than we started with. So as we've had also, I think what's helped us maintain our enthusiasm is the development of new analytical tools in gene sequencing, gene expression, microbiome research, which is not new per se, but is relatively new. So these offer new opportunities for us to delve deeper and look at more mechanistic aspects of the work.
Yeah, thanks. That makes sense. And I would guess that another aspect of the energy level and enthusiasm is that you're constantly bringing new people onto the research team. And in fact, one of our attendees asks, who did your research team involve at the university? And were there junior faculty and students among your colleagues working on these various projects?
Absolutely. When we started 20 years ago, I guess I could have been called a senior research person, but I didn't see myself at that point. I was in mid-career and brought in a number of people who were recent PhDs. Those people now are at mid-career, and we're continuing. We have a fairly strong cadre of graduate students coming in from all over the world that bring their own series of problems and perspectives to our program. from Asia, from Europe, from South America, from Africa, and the subcontinent of India, Nepal, and so on. We even have some from the Mideast. All of these people come to us, and interesting, from my perspective anyway, is that they often ask very challenging questions. And so this has energized, I guess, our program, and we encourage, we have journal clubs, we talk about this stuff a lot. So we do have, I guess, a whole range of ages or people at different stages of their career. And my job, I guess, in part is to see that they develop skill sets that will enable them to basically take over and become the leaders of the future, both here in the US and elsewhere.
And I imagine that you make these connections with partners and new members of your research team in a fairly organic process, simply by publishing your work, attending conferences, networking, and people learn about your team and your institute and approach you about the possibility of joining this effort. But I do have a question from an attendee about how it is that you connect with your partners, and whether you have any advice for researchers developing partnerships that can really move the research forward. It sounds as if bringing in these different perspectives raises ideas or strategies that you hadn't previously considered, and that in itself moves the research forward. But can you talk a little bit about how you make these connections and any advice that you have specifically about developing partnerships?
When I came here 23 years ago, I started a laboratory in southern Idaho that involved two people, me and a guy they were met at the university who they transferred down here to get him out of up there. We thought really a lot about this, and two things struck us. Number one, we don't want to go compete with existing laboratories. We need to come up with some new approach that provides value to people that would want to invest in us. And secondly, new technologies. When I started, genomic work was just in its infancy, and we identified that as a tool, a set of tools that could ramify across the spectrum of the activities we're engaged in, whether it be fish diseases, fish nutrition, fish physiology, and so on. How did we connect with people? We worked pretty hard, not only attending conferences and making speeches, but attending other venues, publishing our work, not just in scientific publications, of course, which we have to do as academics for our professional development, but also in other venues. I wrote a column in a trade magazine for 12 years, for example. So we made a big effort to not only connect with our local industries, the regional industries here, but to connect with other entities. And we've been kind of lucky in a way in that aquaculture's growth has attracted the attention of some fairly major companies in the agricultural world who now come to us to say, we have some products, we use them in other applications. We want to know if they'll work in fish. What can you do to help us? And then I'd say the last thing that's helped us a lot has been the University of Idaho's attitude toward industry partnerships. They've been very open and very accommodating to the intellectual property concerns of some companies that come to us, and we help develop products. So I guess it's been a combination of factors, all of which have contributed to our success.
Great. And I do have an additional question from a participant. Before I get to that, though, I'd like to stay on the subject of partnerships a little bit longer and return to one of my seeded questions, my previously prepared questions, which seems relevant to this discussion of partnerships. And that is, what advice would you give to a faculty member who's initiating a HIBAR project in terms of making sure that their partners are adding value intellectually and are truly engaged in the fundamental research, as well as just interested in the final outputs, possibly in commercial products? So again, how do you make sure that your partners, whether they're tribal partners, government partners, industrial partners, are actually involved in the intellectual aspect of your research?
That's kind of an interesting question, because my experience in academia, and I had some few years in government, was that top-down research doesn't get you there. You really have to grow these kinds of relationships organically through sincere partnerships and sincere interest and common goals. I guess communication, listening, thinking hard about and being able maybe to articulate back to your partners, not just what they hope to achieve, but how you can add value or explore dimensions that they don't have the capacity to do or haven't thought of before. So it really, I think, boils down to communication, getting to know people, spending a lot of time doing so. And it doesn't always work. We've had partners who we do some work with them and they say, okay, thank you, see you later, we're done. Others who get engaged and want to continue on. To give you an example of that, we were approached by some pretty big companies I probably shouldn't mention, but big, who were interested in biofuel production and got a ton of money from the Department of Energy to grow algae to produce biofuels, which of course you're probably familiar with. Now, the big question for them was, once we take the oil out, what do we do with the rest of the stuff? We have a lot of algae, protein, and other material we can extract. How can we create value? How can we find a home for that? And the material flow of this, how can we get rid of it and make money, basically, to put it bluntly. So some of those efforts were not that, they did, let's put it this way, the algae products that we explored didn't turn out to be particularly good feed ingredients, in part because algae are tough. They have tough cell walls. They're hard to digest without disruption. The disruption technologies cost a lot of money. It costs a lot of money to dry the stuff. The ways in which they add flocculants to coagulate or precipitate the algae, facilitate a harvest from ponds, turned out to be detrimental in fish diets. So we do have some dead ends that we come to, but that's a value to these companies too, because they know that's something they don't want to explore more, and they can look for other uses, either industrial uses or perhaps feeding to ruminant animals where these issues might be minimized.
Yes, I think it answers the question. And also, it sounds that rather than simply having a few highly targeted partnerships in mind, you make overtures and you make overtures to a variety of different kinds of partners, and you welcome interactions with a wide range of potential partners, and you understand that some of these may have durability and become long-term partnerships, and other times these will be much briefer interactions that may or may not have obvious successes attached to them. So that basically just being open-minded about a wide variety of potential partnerships, including ones that you may not have anticipated.
Yes, and I think another thing to add to that is to be honest with our partners or the potential partners and say, look, your product has problems. We'll try to evaluate them, but before you spend a lot of money trying to commercialize a product, let's do some basic research to see how it performs in laboratories and what potential problems may emerge and what technologies may be employed to overcome those problems, and not try to sulk them, basically. And we try to work and understand their problems and try to solve their problems. So there's a lot of listening involved, and then trying to articulate back our understanding of what their objectives are, and so that we're pretty much in concordance when we start these projects.
Good, thank you. And speaking of negative issues, we do have another question from a participant regarding your final slides, in which you noted some negative societal impacts of certain types of aquaculture. And our attendee asks if you could say a bit more about how you vetted the research in advance to gauge possible public concerns, or more specifically, is there an accompanying integrated public outreach element to your research program?
Well, to answer the second part of your question first, we've been engaged in some, or gotten engaged anyway, in highly controversial issues in the Pacific Northwest concerning salmon farms. And we try to avoid being perceived or being cast as being advocates. To give you an example, I was invited many times to meetings in the Monterey Bay Aquarium or with Seafood Watch or some of these other entities. And they would say, well, we want somebody down here who's an advocate of aquaculture. And I would say, well, that's not me. I'm an advocate of facts. And if the facts lead one way or the other to support your contentions or to not support them, that's not my job. So to be honest up front and say, I'm not in the advocacy business. I'm in the fact business. That's my role. You take the facts we develop and then you use them the way you see fit. I don't think we've made great efforts to be highly publicly visible. I did for a while. I mean, I would be interviewed by the Washington Post, New York Times, and for a while I took it upon myself to write and speak on radio, TV shows, and that. To when some of these issues became pretty hot, maybe 10 or 15 years ago, but I got kind of tired of it because it struck me as, and I don't, this is how the news business is. When a plane lands safely, nobody cares. So they're all looking for controversy. They're looking for things that will attract viewers. I didn't find that to be very productive. What I found was be more productive was to begin to engage with some of the people who had different points of view or I felt could value from our research. But what I learned at the end of the day was that some of these controversies are not solved by simply the educational deficit model, which is, I know more than you, and if I tell you what I know, you'll believe what I believe. There is a lot of value perceptions, or a lot of value-driven perceptions of various social issues, and that's hard to change. So I decided that's really not our role. We kind of pull back. We will engage when asked, and we'll be honest suppliers of information, but we kind of steer clear of that unless there's a good reason for us to become involved.
Thank you. And I have one last question before I'd like to hand it back to you for a few minutes of final takeaways. And this question is about funding. How do you maintain a long-term research program like yours with a variety of funding sources, making sure that you have a continuous stream of support for your institute and for your various projects? Do you have any special advice or thoughts about how to secure funding for this kind of HIBAR research for an extended period of time?
The way we've been successful over 23 years, and by successful, I mean we haven't gone out of business. We're not making profits, is to try to identify several long-term consistent revenue streams. One of them being our partnership with the USDA, so that supports our selective breeding that I described. Another being we had some other long-term supports from other government agencies, some of which have dried up in the meantime. And we supplement that with other grants and contracts that we go for. For example, I'm involved in a 5-year NSF grant right now. So we compete in that arena trying to get, those are not, I don't consider those long-term grants, but they're certainly not short-term. And then supplemented with some of the more, the granting cycle these days seems to be limited to 2 or 3 year grants, at least for the agencies we develop. And it's hard to see that how we can develop programs that may take longer to mature and reach their potential with such grants. What we've tried to do is get our team to tackle different aspects of these grants, but have a long-term or a larger vision and try to basically put the pieces together. And then we supplement those with some of these short-term industry grants that help us. Some of these grants are for 6 months and they're quite small, but we don't turn anything down. But I think the key has been to have 2 or 3 key areas where we continue to focus and move this money around as we see to be able to keep things going. It's challenging with graduate student funding these days because sometimes it takes a PhD student 3, 4, 5 years to finish. And so one has to be creative and, have them work on some other projects while they're going along. But anyway, it's just basically kind of a funding salad that we try to keep going.
So creativity, flexibility, and diversification,
Exactly.
what you're describing. So Ron,
Yes.
we have about 2 minutes left, and I wondered if there were any key takeaways specifically related to HIBAR that you'd like to leave us with before we end our webinar this morning.
I guess the takeaway I would have was I didn't know I was doing HIBAR research until I learned what HIBAR research was. But we've always felt here in our lab that we need to drill down to really basic mechanistic aspects, going from the genes to, in fact, even epigenetics, up the pyramid, so to speak, to the organ, tissue, whole animal, and population level. That, coupled with our partners who have been able to contribute quite a bit to our program, not just in their intellectual contribution, but in their ability to buy equipment that we maybe couldn't afford. In our laboratory here, for example, we meetings every so often and say, what do we need? We created a system here where nobody all the labs are commonly used and shared. So we all share equipment and talk about what our highest priority needs. So it's been kind of a team approach. Doesn't always work perfectly. Sometimes, we run into some conflicts, but just to try to have in our agreement, we have built in that as one of the conditions of being a partner with us, that you are going to be a partner and share as much as you can with your thing. But I think we've been very fortunate to jump on the genetic issue early on when it was really not being used very much in not just in population genetics, but also in other applications. So building a strong foundation that can be applied in different ways, I think, has been one of the keys to our success and probably a key to HIBAR research.
Thank you. I think that's a very good note on which to end our conversation this morning. We greatly appreciate your presentation, Ron. I found this very informative and appreciate your time. And with that, I think we're at the end of our hour. So thanks very much, Ron. Okay.
All right. Thank you all.
Okay. Goodbye.